Good. So I'm going to talk to you about the role that the diel cycle might have to understand trade adaptation and biogeography of phytoplankton. So yes, and also I work closely with these people in this work, and Joe and Stephanie are here also. And so we are interested in phytoplankton biogeography. And this is because, for example, we do have some Prochlorococcus or Seminococcus dominated communities in tropics, but some diatom dominated communities in high latitudes. And this is important because a diatom dominated community have much different primary production, much different trophic upgrading to higher trophic levels, or it has also a different impact on the biological carbon pump. And this also facing the climate change, it's important to understand where the different phytoplankton dominate to get better future predictions. And and one of the key environmental factors in these global ocean models is the seasonality of light. And I'm going to show you in a simple model how the seasonality affects phytoplankton. So on the left, you see a figure of the growth rate of phytoplankton at different nitrate concentrations. And we have the so-called gleaner opportunist trade-off. The opportunists used to be like diatoms, and the gleaners used to be analogs of Prochlorococcus. And here, a gleaner used to have lower half saturation constant that gives him better fitness at low nutrient concentration. But an opportunist, like a diatom, has higher maximal growth rate that let him be better competitor at high nutrients. So if we want to have light limitation too, we have a similar, it looks different, but it has a similar growth curve with light. And if we put this simple information in a chemostat model and run the simulation for 2 years, we get this typical seasonal succession with such a simple model that you can have the opportunist dominated during spring because nutrient concentration are still relatively high. And when nutrients are limiting, a gleaner dominates. Now, we know that there are 2 periodic oscillations of light. It's not only the seasonality, it's also the diel cycle. And these 2 figures here have the environmental factors I had in this model with seasonality before. But if we add diel cycles, we get this picture. And these are actually shaded areas, these 2, because we have these fast fluctuations of day-night cycle. So you can imagine this goes like up and down, that's why it's shaded. And when we have more light in the system, we have more uptake of nutrients by phytoplankton. But during the night, we have a buildup of nutrients because usually, in this model, there is no uptake, but usually there is less uptake of nutrients during the night because they grow slower. Now, if we get this information, this mechanism of the diel cycle on top of the effects of seasonal cycle, we favor the opportunists. You can see that it's the same model. Here we just added diel cycles. But here the opportunists are favored by the seasonal cycle plus the fluctuations of nutrients of the diel cycle. And I've run such a similar simple model for different latitudes and nutrient supplies. And here we estimate the competition outcome and biodiversity. And we can see that the impact of diel cycles can be really important, especially at high nutrient supplies, because the more nutrients you have in the system, the stronger the amplitude of diel nutrient fluctuations. So this might be interesting results to support that maybe there is a strong impact by the diel cycle, but it's only 2 species model, it's a simplified chemostat system still, so we wonder if this mechanism would hold in a global ocean setting. And also I want to mention that this mechanism of linear opportunist competition has been shown before. It's not new, like for instance, in this figure from Litzman and Klausmeier, the proportion of light in a day, this is 1, so this is only light, or only light, you don't have dark periods and the cleaner winds in a constant environment. But as you go to an environment that you have half day and half night, as this case, you have the opportunities dominating because of these fluctuations. And I just showed you competition between cleaners and opportunists, but there is a list of traits that might indicate that adding diel cycles in global ocean models might be important to understand more traits and trade-offs on phytoplankton. For example, storage, these fast fluctuations of nutrients. Some people like Gruber, James Gruber, have shown that fast fluctuations favor storage specialists. The circadian clock, we know that it's absent in some species, but present in some other species. The vertical migration of dinoflagellates, that we know that they let the other species compete in the productive layer, but they travel down to get some nutrients and go up to photosynthesize later. And also pigment diversity in phytoplankton might be important during the diel time course, because we probably have something like this diel succession of light colors, because the time that a light beam travels inside the water changes its color. So a phytoplankton community living here probably experience a diel succession of light colors. So all these traits might motivate us to include diel cycles in models. But today I'm going to talk about these two. I'm going to talk about cleaner opportunist competition in a global ocean setting to see if such a simple mechanism holds there. And then I'm going to talk also about storage in a simple chemostat, some preliminary work. So diel cycles in the Darwin model. If you don't know, the Darwin model is a global ocean model of MIT that includes a lot of functional groups and realistic chemistry and physics of the global ocean. And what we do here, we have a setup with 15 phytoplankton, including cleaners and opportunists. We have more realistic conditions like multi-nutrient limitation by nitrogen, phosphorus, and iron, realistic dilution rates compared to a chemostat that you can have slower turnover rates in oligotrophic environments compared to apollonic regions that you have fast regeneration. And what we do is we add diel light cycles and we resolve the model 2 scenarios, one with diel cycles and one without, so we can compare them. Now, this huge panel I have that I'm going to try to focus on some areas. Dynamics of 5 different locations for an annual 10 course. And you can see the locations here. So location 1 to 3 are in tropics, and then location 4, a temperate, and the polar location in 5. So in general, you can see in the diel scenario, like the gray, that when we have diel cycles in par, we have an impact on primary production, then also functional groups fluctuate. And I want you to focus on nutrient dynamics. What you see is that only one limiting nutrient fluctuates here. And if we look on the nutrient limitation in the model that you see here, we have BAN limitation here and iron limitation in location 2. Adam, I don't know how it looks to you, this one. I think it's a little closer to the bottle experiments, but and we don't have co-limitation, we have the law of the minimum in this case. So what we see is that when the nutrient is limiting, like location 1 and 3, then is when it fluctuates. Oops. When it fluctuates like here and there. But in location 2, we have iron limitation. So the result is that under multi-nutrient limitation, the diel cycles are present only in the most limiting nutrient. And that makes sense because in a limiting case, you see that phytoplankton drive nutrients down to the limiting level in the control. And then on top of that, if you add diel cycles, you have some fluctuations. If the nutrient is not limiting, then you have some huge fluctuations based on mesoscale processes of the model or some quick seasonality, even in tropics. But when it's limiting, the nutrient, when it's limiting, it's always in the lower level. And on top of that, you can have a strong impact of diel cycles. Now, at higher latitudes, we see no strong effect, diel effect, because the impact of seasonality is much stronger. So even some tiny diel cycles in this case are not important. And when we look on, so on this plot, I have like the difference between the concentrations we found in the diel simulation compared to the control. So if you see an increase there, it means that when we add diel cycles, we see an increase of nutrient concentrations. And you see that in the tropics that we see before, we have some increases of nutrient concentrations that help the opportunists to do better compared to the cleaner. So that's a main outcome, that diel cycles are able in realistic environments to induce diel cycles in nutrients, and then the opportunists can take advantage of that. And in more, if we focus a little on the different species we have, the fastest growing species, even from the cleaner group, like more subtle effects, are favored. Now I think I'm moving to storage already, yes? Oh, go fast. So now we do see that diel cycles have some large scale effects, and the hypothesis is that these diel nutrient fluctuations might favor some species with storage capacity to take advantage compared to other traits, because these increases of nutrients during the night, probably one species decide to store them and use them together with photosynthesis during the day. And luckily, the Darwin model has a quota version that I switched on, and I can study this hypothesis. However, the results were a bit complicated. I couldn't get a clear picture out of it. And the same happened when I used a simple group model in a chemostat system, in the simplest model with some species competing, some of them to have storage, some of them not. So today, I'm going to just talk to you about trying to understand the group model and how storage could be implemented, and some simple chemostat simulations. So from the group point of view, a cell looks like this. There are some nutrients around, uptaken inside the quota, the Q pool, and then from the Q pool, you make some biomass. And this is expressed with these two simple equations, that the cell approaches the maximum uptake rate at high nutrient concentrations. Also, it cannot grow when the quota is less than a Q mean, but approaches the mu max when the Q approaches infinity in this case. And when we put this in a simple chemostat model, we get something different compared to the simplest I showed you before, the monod model. The interesting thing is that when nutrients are depleted, phytoplankton continue to grow because it uses the reserved nutrients inside the quota. So this is some information that the group model have improved better at simulating this mismatch in dynamics. Now, if we want to build storage in the group model, we should have expressed some trade-off, right? So here I have the simplest, I can think, trade-off of an opportunist and the hoarder. Hoarder, yeah, I think I told you. Yeah, it's a species we call like a storage specialist. So the opportunist has higher growth machinery, but smaller quota, and the hoarder has higher quota capacity and smaller growth machinery. And if we wanted to plot the growth rate as a function of quota, it should look something like this. So the opportunist can grow faster, but its quota is smaller, so it stops sooner compared to a hoarder. However, today I'm going to go even simpler because this is even complicated to understand what is the effect of storage. So I go a step even simpler here, and I have a non-hoarder compared to a hoarder. So these guys have the same uptake and growth rate, but this guy just have higher quota capacity. This is helpful from a modeling point of view, because if I understand this, then I can safely go to a realistic trade-off and hopefully back to Darwin simulation at some point. So this is helpful because the outcome is very easy. So if storage is not beneficial at all in some simulations that I'm going to run later, it means that the hoarder and non-hoarder will have the same biomass if storage is not important. But if storage is beneficial for the hoarder, it will win, it will outcompete in the competition. So try to build this in the Darwin model. Here are the two guys that they have the same growth rate, as I told you. Only the hoarder is better in storing nutrients. So if we want to stop them growing after a Qmax, we need to add this term here, where when Q approaches Qmax, it reaches the peak of growth rate. And if we want to include the Q storage in this growth rate function of the model, we don't need to change this equation. We just need to make a restriction, because now the Q can go higher to Qmax. So when this happens, we need to have a minimum function and say, okay, if Q goes higher to Qmax, take the Qmax in order to have this part of the curve. Otherwise, it would grow even higher than the maximal growth rate of the model, which would be wrong. Now, the uptake part of the model is, which is the simplest version of Droop, is like this. If we want to tell to the species to continue taking nutrients until you fill your quota, the maximum quota, we need to multiply it with this term here, which is with the blue term there on the left. And then when we multiply these two in the model, actually we use this function. And we say, okay, you uptake nutrients until you reach the Qmax. Now to have the storage guy there, we said, okay, now we want you to uptake nutrients until you reach a Q store, not a Qmax. And we change there the Qmax to the Q store. But this makes a problem. And the problem is that if we plot this function, the hoarder is always better at uptaking nutrients, even at low nutrient concentrations. So this is not helpful because we have a non-hoarder versus a hoarder plus gleaner in that case, because it's also better competitive for nutrients. So we need to, what I did to modify this, I use this, looks a little weird function, but they have the same uptake capacity at low nutrients, and when they reach the Q store, they stop uptaking. So now we have a model, at least I have right now one version that I can understand the outcome, and I can have a real non-hoarder versus hoarder competing with each other. So now in a stable environment, you can see, if we want to have this species competing, that when the nutrients are depleted, the hoarder has stored more nutrients that can build a little more biomass during the dynamics. But then they are not out competing each other because they become actually eventually the same species. There is no trade-off on them. When nutrients go low, they have the same uptake. Now I studied this for different, now I add some periodic fluctuations to see if a periodic fluctuation of nutrient can help the hoarder to outcompete. But as I see here, the periodic fluctuations do not help because even in this case with 10 days, although there is some fluctuations in the supply, this is not present in the concentrations, but only on the biomass, because as they are going into the medium, they're immediately uptaken by phytoplankton. And this happens the same outcome at higher periods. So the last thing I have here to show you is that when we have pulses of nutrients, and not in a periodic way, then we can have at larger periods, we can get this result that the storage is important, because during these periods of high nutrients and then limiting nutrients, they have stored more and eventually outcompete the non-hoarder, and the same for larger timescales. And finally, I have a question here. So this is a simulation I showed you before with the pulses of nutrients per day. This is something I want to understand also to put in the diel model. But and when I almost eliminate the storage for the non-hoarder, as you can see there, then these diel fluctuations can help the hoarder to win. So my question is, what is exactly measured as a Qmax in cultures? So does the Qmax only includes nutrient concentration, nutrients that are ready to become biomass really fast? Then in that case, it will be something like this. It will be something like this. And then this would represent lipids or carbohydrates. But if the Qmax includes lipids and carbohydrates, maybe we need to use something like this, like shorter growth rate in the Qmax. And then in that case, maybe the Qmax helps, the storage can help even under fast resource fluctuations. So that's all for me today. A summary that the diel cycles can induce nutrient cycles in the global ocean, and they favor, they increase the most limiting nutrient, they favor fast-growing phytoplankton. And till now, from the preliminary work with storage, I see that the nutrient pulses might be more important for hoarders. Thank you very much.
So we have questions. Adam?
Thank you. That was a very nice talk. I have a sort of a two-part question. The first part is, do we see sort of strong nutrient cycles in the ocean on a daily time scale? So is that observed? And secondly, could you imagine that if there was some flexible storage in phytoplankton, then they would take up more nutrients if it was becoming available, and therefore then there wouldn't be a nutrient cycle or variability on a daily, and therefore they're essentially then out competing themselves. And so would the sweet spot be somewhere where there's a little bit of storage, so that then there's also a little bit of variability? Does that make sense? Like if you have too much, if they have storage, right, then there would be no variability, right? And right, on the diel cycle. And so, do you think that it is an important? So in that sense, I would imagine it would outcompete itself.
I understand. So the first question, if there are some strong diel fluctuations in the ocean, I haven't found anything at low latitudes, and especially in oligotrophic environments. It's even difficult to measure nutrient concentration because it's lower than the detectable level. So if we try to capture some diel cycles, it won't be very easy, I think. I haven't found something, but I have found some studies at higher latitudes that they show that the diel fluctuations of nutrients are very similar to what we found in these global ocean fluctuations. Although the most important effect was at low latitudes in the model, and I haven't found at low latitudes this one. Now about the storage, of course, you say that if you are stored, if you can store, probably the nutrient fluctuations will not be there. But the fluctuation will be transferred to the quota of the phytoplankton. Instead of having them fluctuating in the environment, if you store them when they are available, it will go. So it's actually the problem goes to another. The mechanism is present, but probably in another pool, like in the storage pool. In that case, that's what I'm trying to. I'm working right now with the storage. Thank you.
Heidi?
Thanks. Maybe this is a little off the wall, but I'm wondering if you have either already done anything to look into, or if you've thought of how your modeling might project how these diel cycles in the phytoplankton might be impacting the grazers. Are there diel variations in loss that you might infer from the processes you're seeing in changes in quota, for instance?
How they can affect the grazers, or also the grazers, how they could affect.
Yeah, diel changes in when grazing is happening. Does the model make any inferences about that?
So the good thing of the chemostat analysis I had before, and that I studied the same in the global ocean model, then it was that in the global ocean model we have top-down control. So we saw that the top-down control doesn't, the mechanism still holds when we have predation. Now, how it would affect the predators? Now, the predators usually have slower growth rates. So I wouldn't imagine to see a lot of, I wouldn't think that I will see fluctuations on zooplankton, for example, because of the fluctuations on phytoplankton. Maybe, but weaker. To have some weaker effects on zooplankton.
Online question or if there are any other questions?
You alluded to biogeography, and I apologize if I missed this, but do you have any hypotheses, or do you anticipate that these different strategies would be dominant in different latitudes, for example, or do you have any sort of preconception about where an opportunist versus a gleaner may be selected?
Well, the seasonal cycle helps the opportunists to become better competitors at high latitudes. And now with the diel cycle, I'm thinking that they will help in general the opportunists a little more, but especially at productive environments when you should have stronger fluctuations of nutrients. Now, if storage is important, maybe in apoelic regions, we would have some large phytoplankton that they can store a lot of nutrients. I don't know if I answered your question.
It doesn't sound like you can answer it, but I was just curious to know what your...
I also, big question for me is, we know that all phytoplankton store, but we don't have an idea, I think, where, in what environment storage is important. Is it in a apoelic region? Do we find storage specialists in the tropical conditions, high latitudes? I don't think we have a, don't know if you guys have some idea.
All right. Thanks, Ioannis.
Thanks.